Hello, welcome. This is the accompanying video to the DNA Structure and Function Lab. And what I'm going to show you today has to do with Activity 2. Now, Activity 2 is after you have viewed the video, which was Activity 1, and there is a video link on Blackboard. And now you've answered all the questions to the video. And Activity 2 is what you're doing next. So the first thing it says to do is you're going to start by making a 0.9% salt solution. So I have measured out my 1 cup of water, and I got my 1/2 teaspoon measuring spoon. The procedure asks you to dissolve 1/3 teaspoon of salt in a cup of water. So I've just gotten my Morton salt, and I do not have a 1/3 teaspoon. I don't know that that exists, but that's what it calculates out to be a 0.9% salt solution. So we're not being analytically correct, but if you use your 1/2 teaspoon, and I used my 1/4 teaspoon, and I measured 1/4, poured it into my 1/2, and then I tried my best to do a 1/2 of the 4th and put that in here, and that measured out 1/3 teaspoon of salt. So I stirred it for a few seconds, I let it sit, and you can see the salt settles down to the bottom of the measuring cup. I waited a few more seconds, I gave it another stir, and it takes approximately a minute for all the salt to dissolve. So as it's dissolving, you can gather your other materials. And I just keep going back and giving it a swirl. Now, the next thing is to chew on your cheek for 10 seconds and then sip about 5 to 10 mils of the saline solution. So it's much easier if you get your graduated cylinder from your kit and take your saline solution and actually measure 10 mils. So you have a much more accurate collection of the amount of solution you want to swirl around in your mouth. So I have my 10 mils, and you are to have a small little paper cup, and mine's just a Dixie cup, and I'm going to take my 10 mils and I'm going to pour it into my Dixie cup, and then I can set my graduated cylinder aside. Now I have my 10 mils in my cup. Now I'm going to chew on my cheek gently for 10 seconds. And that's just simply as you would squeeze your cheeks in and kind of gnaw on them. Please don't hurt yourself. You're not trying to make an abscess. You're just loosening up and helping to deflect more cheek cells from inside your mouth. Every single moment of your life when you're talking or eating, your cheek cells, inside your cheek cells, as well as outside on your face, is sloughing epithelial cells. Your clothes, as they rub across your skin, also sloughs off the outer layer of your epithelium. So we're just going to collect some of those, and by chewing on your cheek, you're getting some deeper cells that will have a lot more nuclei and therefore some DNA. So now I'm going to I've chewed on my cheeks, and I am going to take my 10 mils and I am going to swirl it around. You're not to swallow the saline solution, you're just swishing it around in your mouth. And the instructions say for about 10 seconds. The longer you do it, the better your results. So I always encourage my students in class to do this for 30 seconds. So here we go. We're going to swirl, and I am going to lessen the time in the video, but you're going to do it for 30 seconds. You can tip your head from left to right, and then when I'm done, I have simply spit the solution back into my cup. Now I can take my test tube and I am going to pour that 10 mils into my test tube and discard my Dixie cup. And so now I have my test tube in my cup, and I'm going to discard the other thing. So now that I've done that, I have my contents in the test tube, and I want to add 4 drops of liquid soap or dish detergent. Now, you want to make sure you've read the intro and you understand what the purpose of the soap is. And everyone knows that our cell membranes, excuse me, are made up of phospholipid bilayers. And that's oil, right? Lipid. And what do we do when we clean our dishes to get rid of oily, greasy foodstuffs? We use soap. So the soap, the purpose of the soap is simply to break apart the cell membrane. Once we break apart the cell membrane, we will break apart the nuclear membrane, because the nuclear membrane is made out of the same phospholipid bilayer. So I'm going to take my test tube and I am going to add 3 or 4 drops of soap, and it's hard to get drops particularly, so I'm doing the best I can, which is exactly what we hope you'll do. My drops are kind of big and they hang on there, so there I've got my drops, and you can see that the green soap is in the bottom of the test tube. Now you're going to take the parafilm out of your lab kit, and the parafilm is in a little box, or I'm sorry, in a little bag, and it's plastic, and it has a wax. It's like a piece of wax paper, plastic wax paper, and it's got a piece of paper over it that says parafilm. You don't want the paper portion. Throw that away. You want the plastic waxy portion. And if you will just set it over your test tube, put your thumb there, take it and bend it and allow it to grab onto your test tube as such, then I always put my thumb over the test tube, and then we're going to ask you to simply rock your test tube back and forth, making sure you're exposing all those cells to the soap. So this is probably the hardest part, because you want to take the test tube and shake it. But you don't want to shake it, because that will break your DNA into little tiny pieces, and it doesn't look near as cool as the long strings that we associate with DNA, and you saw in the previous video from activity 1. So I'm just rocking my DNA. I'm trying not to make any bubbles. You're going to make some bubbles. But the idea is simply expose as many cells to that soap as you possibly can. The instructions tell you to do this for about 2 minutes. And so we'll shorten the video so that it's not 2 minutes, but you'll do it for 2 minutes and you'll get a whole lot better results. So rocking away, and once you're done with your 2 minutes, you take your parafilm, dispose of it, and I'll set my test tube back in my big cup. And the next step is that you're going to take your 70% isopropyl alcohol. Well, I've kept my alcohol in the freezer the whole time I was setting this up, and I only got it out just now. It's 70%. It says isopropyl alcohol. Some of you may have found it's called rubbing alcohol. You can find this at any drugstore, grocery store, CVS, Walgreens. You can say this was American Fare. I don't know, this was from Rite Aid, so that tells you how old it is. It's isopropyl alcohol. And you need, it tells you, approximately 3 mils. Well, since I did 10 mils for sure of salt solution, I'm going to go ahead and add 5 mils. If you only feel like you did 3 or 4 mils of the salt solution, then you only need, or 5 mils, then you only need 3 mils of the isopropanol. But I'm going to do 5 mils since I have 10 mils of solution. So you're going to take your test tube and lean it. I'm trying to get a good angle here, at a side angle, and you're going to drizzle the isopropanol down the side. And again, the whole reason to do it slowly down the side is to prevent turbulence so you don't break apart your DNA. Now, once I do that, you will notice that, I'm trying to see what is the best thing that I can put behind this. You should see an interface between water and alcohol. They do not mix. And whether or not you can see it on the camera so good, I'm not sure. But hopefully in your kitchen, you can see there is a distinct interface between the volume of alcohol and the water. The alcohol is on top. DNA is not soluble in the alcohol. And so right at the interface is where the DNA will start forming. And so the easiest thing for you to do is set your test tube in your cup and let it sit for approximately 1 to 2 minutes, and you will see DNA. One of the easy ways to see it is you're looking for bubbles. There'll be bubbles that form, and those bubbles will actually hang on to a strand of your DNA and be pulling it up like a balloon, as if the DNA is the string and the balloon is rising to the top. So right here at that interface, I'm beginning to see bubbles, and you should begin to see your DNA. And at that point, remember, you can take a picture. That would be a good thing to do. And then you can get your little stick. There's a little wooden stick in your kit. And with the wooden stick, you can spool the DNA. And right at that interface, it's the wooden stick is made out of wood. It's polar. And the DNA likes the stick. And so if you carefully, slowly put the stick right there at the interface, the DNA grabs onto the stick, and then you can twist it so the DNA will spool around your stick, and then you pull it up, 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 and as it's coming out of the water, you may see a little string. You'll have a lot more DNA than I, and you can even look at your stick, and it looks basically like snot, and it'll be on the end of your stick. So that would be, and of course, take a picture. That would be how you are going to collect your DNA using some simple things from home. So remember to take your picture at 1 or 2 steps during that whole process, and you will be good. And then you can answer your questions after you've completed activity 2. Now, remember, activity 3 is not there. We deleted activity 3 as it is done in class with material in class that we don't have the ability to put in the kit. So we're moving on to activity 4. And activity 4 is about transcription of DNA to RNA. So transcription is taking the sequence of nucleotides from DNA and converting them into nucleotides, making a messenger RNA. Now you have papers in your kits that you are going to cut apart. And so I have like the yellow sheet. I have cut these all apart on the solid lines, right? And that has a D on it. These all have a P, and I have groups that have G or a D, or I mean a G or an A or a T or a C. Each one of these is a component of a nucleotide. So the D stands for deoxyribose, of course. That's the sugar in DNA. And I'm going to group all those together. The P stands for the phosphate, which is the component of a nucleotide. And then here is the base. And like I said, we have G's and C's and A's and T's. So these three components are what you're going to use to build 9 DNA nucleotides. Now, if I've cut these apart. The D, of course, stands for deoxyribose, which is the sugar, the 5-carbon sugar. And what's good about this tool is that the first carbon here is labeled 1'. This is the second carbon. The third carbon is 3', 4, and 5'. The prime, the little apostrophe there, is simply a tool that we use in organic chemistry. The nitrogen base is considered the most important functional group on the entire molecule of a nucleic acid. All of those carbons are also numbered. For our purposes, we don't need to know that. We're not going to look at the carbons in the nitrogen base, but they are numbered. In the sugar, so deoxyribose in this case, the sugar is also have a lot of carbons, and so we're going to number those carbons because it's an incredibly important component. And so that's where the 1, 2, 3, 4, 5 come from. There are 5 carbons in a nucleic acid sugar. And often it's referred to a pentose, and often someone just says the 5-carbon sugar of DNA. The number 1 is telling you where the most important functional group is on that molecule. And so the most important functional group on this deoxyribose is the nitrogen base. So the nitrogen base is attached to the 1' carbon. So we made a line to indicate that there is a bond there, and the nitrogen base, it doesn't matter which nitrogen base, every nitrogen base in a DNA molecule is attached to the number 1 carbon on that deoxyribose sugar. Then as you move around, you see the 3', and the 3' either has a hydroxyl group, which is an OH group, or it has the next nucleotide. It's holding the next nucleotide. The 5' sugar is the one that I tell students in classes represents the chimney on this house. The oxygen is the roof of the house, and then there's a chimney running right next to it, and that's the number 5 carbon. 1, 2, 3, 4, 5. And the number 5 carbon is holding the phosphate group. And again, we have a line there so that you can see there is a bond holding that phosphate group on there. And we've cut them trying to imitate a puzzle piece so that you can only put them together in one way. So the phosphate goes on the 5', and it's cut like a roof of a house. And what you're going to do is when you take apart the stacks, right, all of the P's and all of the D's and all of your bases, you are going to build nucleotides. So a nucleotide has a sugar and a phosphate on the 5' carbon and a nitrogen base on the 3' carbon. You're going to tape the nitrogen base onto the sugar and the phosphate onto the sugar. So that once you have these taped, you will have one nucleotide that looks like that. Now, it's not important that it's perfect for you. I feel quite certain that you can tape and cut these things apart. But what you want to do is use this as a tool to realize this nitrogen base is attached to the 1' carbon and the phosphate is attached to the 5' carbon. So this is one nucleotide. So you're going to do that with all the others. Here is a second nucleotide. I put the phosphate on the 5' carbon and the adenine on the 1' carbon. So I've got guanine, adenine. Here's a cytosine, and I've got it taped together. This again is another nucleotide. Here is my fourth nucleotide with thymine. Noting the 1' holds the nitrogen base, the 5' carbon holds the phosphate in every single nucleotide. So that's what in activity 4, as you went through your instructions, step 1 here is telling you to group the paper pieces that you've cut apart into the individual groups, all the D's, all the P's, and then the nitrogen bases. So step 2 tells you to build the 9 nucleotides. And indeed, you have all of that material available to you to build 9 nucleotides. Do not tape the nucleotides together. You want to be able to manipulate the nucleotides as we go through the activity. The only thing you tape together are individual nucleotides. Now, you have these things on your desk, and so it says you're in step 3 now. Orient the paper DNA model in such a way to construct a single strand of DNA with 9 nucleotides following this sequence. So it tells you to start with T, and I'm going to put T all the way up here at the top because this is going to be long. Then it is followed by an A. And one of the first things you realize is that when you're making a strand of nucleotides, there's the 3' carbon on the sugar. It bonds with the phosphate of another nucleotide. So in order to put these 2 nucleotides together, the only place they'll bond together is at the 3' carbon and the phosphate of the 2 separate nucleotides, so that you can see easily when you put those down that your backbone on your DNA molecule is phosphate, sugar, phosphate, sugar. So I will continue this and I will make it according to the chart. So it's TAC, and I've got my C, and then there's another A and another C. So I've got this, and I'll just keep adding my nucleotides, making my strand, and it's followed by a T, and then a G, and another G. So I have a nucleotide strand that runs TACACTGG. Don't tape those together. You just are building this nucleotide strand. Now, you can use this on its own as a helpful tool. Is thymine a purine or a pyrimidine? Is adenine a purine or a pyrimidine? Look at all of these, use some information, right? Your purines, here's adenine, you got your 2 adenines, then your guanine, there's your purines, then your thymines and your adenine, your thymines and your cytosine is your pyrimidines. So use these. I know they're not puzzle pieces and they're just paper, but they're a really good resource. You can shuffle them up and practice saying them over and over and over. Now, the bases are pointed up and you are doing transcription. So that means this represents the gene, and now you're going to use your RNA strand. And you would have built your RNA nucleotides in step 2 exactly how you did the DNA nucleotides. So I have the blue paper. I know these are RNA because the sugar is ribose. And ribose sugar, the phosphate is still hooked to the 5 prime, and the nitrogen base is still hooked to the 1 prime. So I have my guanine, I have my adenine, constructed exactly the same way. Notice I did not make this exactly straight. It's okay, but I know that adenine is indeed connected to the 1 prime carbon. One of the differences you noted was there is no thymine when you're making RNA nucleotides. You have a uracil. So the uracil is connected to the 1 prime just like the other bases. And since uracil replaces thymine, you know it is a pyrimidine. Now, you can only match purines with pyrimidines, as you learned in the DNA chapter on structure in your textbook. But you're going to transcribe. So here's thymine. What is the complementary base that will go to thymine? So of course it's going to be adenine. Now I can put this adenine up here and lay it side by side, but you and I both know that the sugar, the ribose, does not connect to the thymine, nor does the phosphate. The only thing that can connect to that nitrogen base is another nitrogen base. And so the adenine and thymine are the complements, and you see between adenine and thymine, even in transcription, there are only 2 hydrogen bonds that form between those bases. And I had to flip my RNA nucleotide upside down in order to orient it correctly. I could have flipped it to get it this way, but we leave that side intentionally blank so that you can't see the molecule that way, and that you have to flip the molecule upside down. So for thymine here, I'm going to add adenine, and I'm going to orient it properly. And then I see my next DNA nucleotide as adenine, and I don't have a thymine in RNA, I have a uracil. So I'm going to place my uracil as the complement to it. And again, it's got 2 hydrogen bonds. Cytosine pairs with guanine. It's got to be upside down, and that's the first time I note that there are 3 hydrogen bonds paired between those 2 nitrogen bases, and it's a stronger bond with 3 hydrogen bonds than just the 2, exactly as it is in DNA double helix. And then finally, here is an adenine, and I would need another uracil, and you continue adding your RNA nucleotides to this strand. And then as you do this, you've noted that the nucleotides are upside down to each other, and that is the anti-parallel directionality of these molecules. If you had a double-stranded DNA, if I take this RNA away, and I want to put, here's a guanine with this cytosine, and make a double-stranded DNA. So if I made this a double strand, I could do this, and you could easily see that I have a double-stranded DNA, and with that double strand, 3 hydrogen bonds between cytosine and guanine, 2 hydrogen bonds between adenine and thymine, and this strand over here is upside down or anti-parallel to this strand. Another great tool that you can use this resource for is to note directionality. So you can see on this side of the strand, the very last sugar, the 3', does not have anything holding on there. And the great thing about using this paper towel is I can actually write, that's supposed to be an OH group. That doesn't look so clean. But there's a hydroxyl right there. And so this molecule strand begins as 3'. Then here's your 5' sugar. And it jumps all the way up here to the next nucleotide to 3' and 5'. The next nucleotide is 3' to 5', 3' to 5'. So this nucleotide strand ends at 5'. It began at 3'. And so typically you see an arrow drawn. Maybe I could draw that with the marker so that it shows up more. And it'll show you the directionality of 3' to 5'. Whereas on this side, it's anti-parallel. So here's your 3', and I will add my hydroxyl group here, noting the 3', 3', 5', 3', 5', 3', 5', 3', 5'. So here is my 5' at this end, and there's a phosphate on that end of the molecule. And this one goes 3' to 5' with the arrow going up. Let me see if I can move the camera so that you can see it maybe a little better. And noting the directionality of the molecule, this can help you learn the 3' and 5' that's necessary in replication as well. This is a double-stranded DNA, and DNA polymerase will be the enzyme that lays down the new nucleotide. So if I remove these, I think you can see those now. So let me become DNA polymerase. Here is my strand, and it's been unzipped by helicase, and it's been unwound by topoisomerase. And now I want to build the new DNA strand. So since I'm DNA polymerase, the only way I will build this molecule is I must find the 3' end. So here's my 3' end. It's the only end I'll start at. If I see 5', I will ignore it, and I will go all the way to the end where I can find a 3'. So I find the 3'. I have the free nucleotide that's floating around in the nucleus. I grab it because that's the complement to A, and I have got to place it upside down to orient it properly so that it is anti-parallel, and I would lay down that thymine anti-parallel, and the 3' is here, and the 5' would be in this orientation. And DNA polymerase is going to do the exact same thing with the next DNA nucleotide, which is a thymine. So I find an adenine. Here's the free nucleotide. I attach it. The phosphate attaches to the 3' of the previous nucleotide. It also runs 5 to 3. So see, I lay down as DNA polymerase, I lay down in a 5' to 3' direction. I must find 3' to get started. Here is a guanine. I must find cytosine. I cannot lay it down this way. I must flip it upside down. The 3 hydrogen bonds form. My 5' to 3' direction stays intact. There's another guanine. I find another cytosine, and I lay it down. And again, it runs 5 to 3. So the new strand that DNA polymerase lays down runs 5' to 3'. Now, you're going to do just the opposite of that if you are going to carry out transcription. So transcription, I'm going to find my DNA strand. It's going to unwind with topoisomerase. It's going to unzip with helicase. And it is going to get ready by putting a promoter in place so that I know this is where I'm going to begin. This is the beginning of the gene. And I am RNA polymerase now. And RNA polymerase finds the 3'. I am going to lay down in a 5' to 3' direction. So here's the adenine. What is the complement to adenine? Can't be thymine. I'm making RNA. So I am RNA polymerase. I must grab a uracil nucleotide that is floating around in the nucleus, and I lay it down. The next nucleotide I read is thymine. I find an adenine. I lay it down anti-parallel in the 5' to 3' direction, aligning my 3' with my phosphate of the new nucleotide. Next nucleotide is guanine. I flip the free cytosine nucleotide. I make sure that the phosphate of the 3' of the previous nucleotide attaches, and I have this going in 5' to 3' direction. And then I look at the next nucleotide, which is guanine, and I lay it down anti-parallel 5' to 3'. So my transcript has been laid down 5' to 3'. I only read the DNA in a 3' to 5' direction. So that should help you as you put your transcripts together. You're going to continue to follow the information. Here's the dashes, and the instructions tell you to delete the dashes and add in the correct nucleotide bases. There is an image of how your nucleotides should look, and then you can just and then take a picture at the end of your transcript so that I can see that you were able to carry out the activity. Then when you get on to translation of RNA to proteins, that's your next step. So you've got this, you've got the instructions from DNA in your RNA transcript, and this thing, remember, this transcript is in the nucleus. It's not ready to leave the nucleus until it makes sure it has the correct exons and the introns are removed. It needs a guanine cap and a poly A tail. Then and only then will it finally go through the nuclear pore and enter into the cytoplasm and find a ribosome. And it'll be there on the ribosome that you will carry out translation. Okay? And you want to make note as you're reading through the text that it talks about control mechanisms of gene expression. And gene expression is what we're doing by turning a DNA gene into a protein. That is gene expression. And there are several, at least 6 steps noted in your book about the ways that the cell controls gene transcription. Gene expression. And transcription is the #1 way, even so much as back when the DNA is supercoiled. If it's supercoiled and tightly bound and wound up in itself, the enzymes cannot get in there, and RNA polymerase itself cannot read the individual letters to pair up the complement. So transcription is the #1 way that cells controls expression. It's the most common way that it happens. So you're going to carry out your translation of RNA to proteins, and you're going to use in your book, you just keep following. You're going to copy your codons from your messenger RNA that you made here, and then go on to use your genetic code to build your protein. So you can do that with this activity and then move on to activity 6, and you're going to do the same thing with an actual mutation that occurs in many people with just one letter out of place, creating sickled hemoglobin molecule inside red blood cells and creating sickle cell anemia. So you'll do the exact same steps, but you don't have to use your paper models, but they are very helpful, and I would encourage you to use them and write on them as I did on the paper towel. Use a tool like this and do it as many times as you need to so that you can get comfortable with transcription and translation. I've also posted 2 worksheets in the activities portion on Blackboard. And the snorks from outer space are a tool that is very helpful in learning translation. So hopefully this helped, and I will post the next video for the next lab in the next folder.